Hey everybody, welcome to another bonus episode of Molecast. I am Mole. Welcome and happy Wednesday or Thursday, depending on what time zone you're listening to. And if you were around, I don't know, maybe 12 hours or so ago, you might have heard an excited shout in the distance. That was me, because the Nobel Prize in Chemistry was awarded today. And biochemists rejoice. Not only was I excited because two women were awarded the prize, but it's all about CRISPR-Cas9, and I'm so excited. I'm imagining the meme with Jonah Hill, and he's just like excitedly going, ee! That's pretty much what I did this morning. I was so excited to see that this body of research was recognized and awarded by the Nobel Committee. And so today we're going to talk about what is CRISPR-Cas, what is the CRISPR-Cas system, why was it awarded, and why is this such a huge deal for both biology and chemistry. So let's go ahead and get started. The awardees were Dr. Jennifer Doudna of University of California, Los Angeles, and I'm really sorry, I'm going to butcher this name, Dr. Emmanuelle Charpentier. And she is from the Max Planck Institute in Berlin. So what these two researchers did was they created new technology, which essentially is able to cut and paste genetic sequences of DNA of animals, plants, and microorganisms with extremely high precision. So think about just the copy paste that we use on the computer every day, whether it's copy pasting text into other documents or files that we have, copy pasting items into Photoshop, Illustrator, PowerPoint, etc. Being able to take that idea and using it for genetic sequences. So just getting everybody oriented, because a lot of Molecast listeners are not scientists. When we think about DNA and RNA and proteins, this is the basic dogma of biology. And we can think about DNA, RNA, and proteins as being a music score, where DNA is the original music score. Like, this was handwritten by the composer. Only one copy. You don't want anything bad to happen to it. So what happens is that publishers then make copies of the original score. We think of those copies made by the publisher as RNA. And let's say within those copies made by the publisher, you only need one particular part. Let's say either first violins or viola or French horn. That is considered the protein, because from the copies that we get from the publisher, we can highlight different parts that we're interested in, whether that be a solo part or a particular group within the entire ensemble. So from there, what is actually going on when we think about CRISPR is, if we go back to the original score that was made by the composer, and we look at the score and we see that maybe the composer had a really late night. Maybe he had, just a random thought in his mind. And as we're reading through the score, there's a region of music that doesn't make any sense. We're in the key of D major for, the entirety of a movement. And then all of a sudden, we see E flat major. Why are we going in E flat major? So what CRISPR's able to do is it's able to go into that score and remove and add new sequence of DNA. In the analogy that we're using, it's removing parts of music that don't make sense within the framework, and adding music that does make sense. So copy pasting the original score, which then allows us to make copies with the correct changes that we've made, and then highlighting the different parts with proteins. So this is huge. It has a huge impact on life sciences. It's able now to contribute to new cancer therapies by editing immune cells in order to combat the cancer. And it may make the dream of curing inherited diseases come true. And the background as to how CRISPR-Cas systems came to be was during Charpentier's studies of a bacteria known as Streptococcus pyogenes. This is a harmful bacteria. She discovered a particular kind of copy in the bacteria known as tracer RNA. And what tracer RNA was able to do for the bacteria was it was a part of the bacteria's immune system or defense against other viruses that could come in and infect the bacteria. And what tracer RNA did was it disarmed those viruses by cleaving their sequence. And this study was groundbreaking, and it was published only 9 years ago. So if you've been following along with our bonus episodes, you've been hearing me say, this research has taken 50, 70 years. This was only 9 years ago. 9 years ago, I was in college. I was a sophomore in college, finishing like my freshman year, just beginning my interest in science. So this is how long ago this was. This was not long ago at all. Then after the study was published in 2011, Charpentier collaborated with Dr. Doudna, who is an experienced biochemist, and she has a vast knowledge of RNA. I've heard one of her talks in person. She is an amazing scientist, also a lovely person, just to hear her chat with. And what they were able to do together was they were able to reprogram this machinery so it would cut any type of DNA, not just viruses, at a predetermined site. So I kind of gave an analogy of how CRISPR-Cas could work in a musical context. But what actually goes on in CRISPR is that in a scientific context, it's actually a region of DNA with two distinct characteristics: the presence of a nucleotide repeat and a spacer. And the repeated sequence of nucleotides are distributed throughout a CRISPR region. If we go back to our music analogy, let's think of this as a particular part, let's say the timpani, that's making a constant beat. This is our repeated set of nucleotides. And then the spacers are essentially bits of DNA that are interspersed along these repeated sequences from the virus. So this is the mistakes that are currently going on in the music, back to our original analogy. So putting this all together, we have a constant beat from the timpani, and then between those beats, some musical context or musical line that's in E flat major, not D major. So then when the virus will attack again, a portion of this sequence is transcribed and made into an RNA. So going back to our music analogy, when we see the E flat major come up, what happens is that we then make a copy of that sequence, and that sequence is known as a CRISPR RNA or a crRNA. So then we now have a third player that comes into play, the Cas protein. And in the context of DNA, we know it mainly as Cas9. But what I want to make sure that we all understand is that there's many different types of Cas proteins. And Cas proteins don't necessarily have to edit DNA. They can actually edit RNA, both single-strand and double-strand RNA. And you can engineer Cas proteins to recognize particular sequences. But in the context that we're going to talk about today, we're going to talk about Cas9. So Cas9 uses the CRISPR RNA as well as the tracer RNA that I talked about before. And it essentially guides where we need to cut in the sequence. And finally, there's a safety mechanism. So we don't want this Cas9 and these two RNAs to cut anywhere, because first, that would be chaos. Even though we know chaos well, we don't want to cause unnecessary damage. We want to be able to cut exactly where the issues are. So what's found in the sequence is something called a PAM motif. And if you don't have a PAM motif, you do not cut. So going back to our music analogy, if we have, let's say, an editor go in with these copies that they made from the score, and they see this PAM motif, or in the case of music, let's say they see a particular D major arpeggio that goes into the E flat major sequences. So you have D major arpeggio, pound, E flat major. If the editor sees that, he knows, oh, this is a region of the music that I have to remove. So this is what's happening in bacterial cells. So what Doudna and Charpentier did was they actually fused the CRISPR RNA and the tracer RNA together, and they called this new RNA a guide RNA. So when I or others manipulate cells with CRISPR-Cas systems, we use guide RNAs. And we use that to cut the DNA and remove regions of the DNA that we do not want. Once the DNA is cut, then what happens is the cell's natural repair system will kick in to repair the DNA. And during this repair, you don't necessarily have to remove DNA. You can add new sequences of DNA back in the original sequence. So for example, back to our music analogy, let's say the editor removes all of the parts of the music that are in E flat major, but he wants to put in new parts that are in D major. So as he's gluing the music back together, he could add in those parts that are in D major. In human cells, in science, usually we use this system either to introduce mutations that could represent different disease types, or we're trying to initiate the production or loss of a protein that's being produced by the natural machinery in the cell. So this is huge. I personally use CRISPR-Cas9 in my system. I am finishing up my research right now with my PhD studies using a cell line that we created using CRISPR-Cas9. CRISPR-Cas9 is something that has completely revolutionized the field. And the big question now is, where do we go from here? Not only as scientists that use CRISPR-Cas9, because you can use it in virtually any system, plants, animals, bacteria, even viruses. But where do we go now? The hope is that we can start using CRISPR-Cas9 to treat neurodegenerative diseases. However, there's also some tough ethical questions that face the field. Some of you may or may not know that in 2018, a Chinese scientist was charged with using CRISPR-Cas9 to edit fetuses that were then brought to term and born. That scientist was charged not only by the Chinese government for 3 years, but also ostracized in the scientific community. There have also been questions as to who owns the technology regarding CRISPR-Cas9. Some of you may know that there's currently a patent battle within the U.S. Patent Law Office of 3 scientists that are currently disputing who owns the technology. Doudna, Charpentier, and another researcher from Cambridge, Massachusetts. So the questions now that really face the field is, where do we draw the line as to how we use CRISPR-Cas systems to improve human life? And when do we decide to use CRISPR-Cas9 systems to improve the quality of human life? These ethical questions are things that are currently ongoing in discussions within the scientific field. After the 2018 studies came out, Doudna, Charpentier, and many researchers expressed their dismay and disgust that CRISPR-Cas9 and CRISPR-Cas systems were used in this way. However, this is something that's currently ongoing, and we're not really sure as of this time how it can be used. Also, CRISPR-Cas9 in non-ethical hands can be used for more malintent purposes, such as editing sequences of genomic material that could cause disease and cause harm to not only humans but also ecosystems as well. So we're trying to deal with these ethical questions and as we go into a new decade, because again, like I said, CRISPR-Cas9 was discovered only 9 years ago. It hasn't even been a whole decade yet that we've had CRISPR-Cas9, which is first just revolutionary for the Nobel Prize in chemistry. Most chemical studies that are awarded the prize usually have been around for at least 2 decades. Last year's winner was 89 years old, and he was awarded the prize along with 2 other researchers for their work developing lithium-ion batteries, the same batteries that we use in phones, laptop computers, etc. So this is a very sharp contrast to last year's prize. However, looking at some of the positives that have come out of awarding this research, Charpentier did not expect to find tracer RNA. This was an unexpected discovery. And this really hits home with the fact that sometimes the things that you try, that you're aiming for may not work out, but you'll find unexpected discoveries along the way. It's not as common in research, but when they do happen, Charpentier trusted her gut and she went for it, and it paid off for her in a big way. Also, both Charpentier and Doudna are the 6th and 7th women to receive a Nobel Prize in chemistry. Previously, only 5 women received an award. So this is another huge step for women in STEM. We've all said multiple times on the server that 2020 has been a really tough year for all of us. But I feel like the Nobel Prize committee and the other committees that are involved in nominating and awarding the prize have really helped take huge strides for women in STEM. So I applaud them. If we only have one, small blessing this year. I mean, we've had a few, one being the servers that we've made, but if this is another small nugget of good, it's that women in STEM have taken a huge step forward this year with all the awardees in both physics and chemistry. Fun facts: each winner, Charpentier and Doudna, will receive $560,000 US. And I found this really funny. Doudna received a free parking space on UCLA's campus. That is priceless. Who needs the $560,000 US? You got the free parking space. If you're in college or you're in academia, you know that those parking spaces are priceless. So congratulations for your free parking space. Hopefully they also put up a sign that says this is your parking space. Because I know, I do not have a car. Chicago is not conducive to having a car. But I know of some graduate students that do have cars, and they complain all the time that finding a parking space is impossible. So congratulations on your new parking space. So yeah, that is, parking spaces are priceless. That's really, the Nobel Prize, instead of awarding people money and a lovely medal, they should just give people parking spaces. That is the Nobel Prize in Chemistry. I am super excited that it's, something that's near and dear to my heart, being a biochemist. The last thing I wanted to mention too was that disregarding the award that was awarded last year for lithium-ion batteries, there's now a new trend in Nobel Prizes in chemistry going to more biochemical studies and processes of life in molecular biology and biochemistry, which is really interesting. But just kind of what Justina said yesterday, a lot of the technology that we currently have now is allowing us to make these major discoveries in biochemistry and molecular biology, since they're both closely related, that allow us to not only understand how life works and how cells work, but allow us to treat diseases in new and unconventional ways that maybe diseases couldn't be treated before. And I found that really interesting because we usually think of the Nobel Prize in physiology or medicine to be a field where maybe biochemistry or molecular biology would be highlighted. But the Nobel Prize in chemistry has really seen the advances that biochemistry has taken throughout the years. And we're obviously, as a society, benefiting from those advances. So I just saw that in my notes, and I wanted to make sure that I mentioned that to all of you. So that's it for tonight's bonus Molecast episode. Tomorrow is the Nobel Prize in Literature. I know this is the science server, but because this is the month of Nobel Prizes, we're going to highlight all of the prizes, because not only are we scientists, but we're also musicians, music lovers. Some of us are fans of literature, others are fans of economics. So we want to highlight all of those different awardees here. So tune in tomorrow, again, 7 p.m. Eastern Standard Time. Sorry, EU gang, take it up with the Nobel Committee. They need to award their prizes earlier. I'll see you guys here as we discuss the winner of the Nobel Prize in Literature. So thank you guys so much for tuning in, and hope to see you soon. Bye.